Today we are comparing two popular plasmids used in molecular cloning, PBR322 and PUC18. First up, let's talk about PBR322. It's one of the earliest and most versatile plasmids out there. It's known for having two antibiotic resistance genes, one for ampicillin and the other for tetracycline. This allows for dual selection. You can easily identify bacteria that have taken up the plasmid using these antibiotics. PBR322 also has a lower copy number, which means that it can produce fewer plasmid copies per cell. It is perfect when you need to clone genes that might be toxic in higher amounts. On the other hand, we have PUC18. This plasmid is smaller and simpler with a higher copy number, making it ideal for producing large amounts of DNA. It only has one antibiotic resistance gene for ampicillin, but its real standout feature is the blue-white screening. PAC18 has a lag-Z gene that allows you to quickly distinguish between the recombinant and the non-recombinant colonies. If your DNA insert disrupts this gene, you will get white colonies on X-cal plates. Here is a more detailed and in-depth comparison of the two plasmids, highlighting their key features, differences, and applications. PBR322 has a low to moderate copy number, producing fewer plasmids per cell. This is ideal for cloning genes that might be toxic if overproduced. On the other hand, PUC18 has high copy number that yields large amount of plasmid DNA, perfect for high yield cloning. PBR322 is larger, around 4.4 kilobases, while PUC18 is smaller, only about 2.7 kilobases. PBR322 has two antibiotic resistance genes, one for ampicillin and the other for tetracycline. On the other hand, PUC18 has only one antibiotic resistance gene for ampicillin. PBR322 relies on the antibiotic resistance for screening, which can involve more complex positive and negative selection processes. On the other hand, PUC18 allows for blue-white screening, where the white colonies indicate successful cloning, making it quicker and easier to identify the recombinant clones. Competence refers to the physiological state of a bacterial cell in which it can uptake extracellular DNA from its environment, a process critical for the genetic transformation. This state can be either natural or induced artificially. In the naturally competent bacteria, specific proteins and machinery facilitate DNA binding and uptake, often during certain stages of growth or in response to the environmental stress. Whereas artificial competence is typically induced in laboratory strains of bacteria like E. coli through chemical treatment, example calcium chloride, or electrical shock, known as electroporation. These methods temporarily disrupt the bacterial cell membrane, creating pores through which the DNA can enter. Once inside the cell, the DNA can be maintained as a plasmid or integrated into the host genome, depending on the experimental design. Competence is the key tool in molecular biology for cloning, gene expression studies, and genetic engineering. Alpha complementation is a molecular biology technique used to detect the recombinant plasmids in the bacteria. It involves the use of the lag-Z gene, which encodes the enzyme beta-galactosidase. The lag-Z gene is split into two fragments, the alpha peptide that is encoded by the host and the omega peptide encoded by the plasmid, such as in PUC18. When a plasmid with an intact lag-Z alpha gene is introduced into a host strain expressing the omega peptide, the two peptides complement each other, forming a functional beta-galactosidase, which turns colonies blue on X-cal or IPTG plates. If foreign DNA is inserted into the lag-Z alpha gene, the alpha peptide is disrupted, preventing complementation and resulting in white colonies indicating successful recombination. Replica plating is a powerful technique used to detect recombinant plasmids by transferring cells from one agar plate to the other in an identical pattern. A sterile velvet pad is pressed onto the surface of the original plate, picking up cells from each colony. This pad is then pressed onto a fresh agar plate, creating a replica. By using selective media like one containing antibiotics, researchers can easily identify colonies that have lost resistance due to successful DNA insertion. The corresponding colonies can then be recovered from the original plate, making this a precise method for identifying and studying recombinants. Imagine you have a stamp that picks a pattern of dots from one piece of paper. Now you press the stamp onto another piece of paper and the same pattern of dots appear. In science, we use a special stamp to pick up teeny dots of bacteria from a jelly plate and put them onto another plate. If some of the dots don't grow on the new plate, it tells us that something special has happened to those bacteria and we can go back to the first place to find and study them. Insertional inactivation is a technique used in blue-white screening to identify recombinant bacteria. In this process, a DNA fragment is inserted into the lag C gene of a plasmid, which normally produces an enzyme that turns colonies blue when grown on a special medium. When the lag Z gene is disrupted by the inserted DNA, the enzyme is not produced and the colonies remain white. So, white colonies indicate successful insertion of the DNA fragment, while blue colonies indicate non recombinant plasmid. This makes it easy to visually identify which bacteria have the desired recombinant plasmid. Replica plating allows scientists to transfer bacteria from an original plate to multiple new plates with different conditions, such as different nutrients or antibiotics. This technique helps in identifying colonies with specific genetic traits like loss of antibiotic resistance due to successful DNA insertion without destroying the original colonies. Insertional inactivation is used in conjunction with these methods like blue-white screening to easily identify bacteria that have taken up the recombinant plasmids. By inserting a DNA fragment into a gene like LAC-Z, researchers disrupt the gene's function, which can be visually detected by a change in colony color. This makes it simple to pick out colonies that contain the desired recombinant plasmid.